Well, thank you, everybody, for coming out on this gorgeous evening. We are thrilled to have you here, and we're also very grateful for those on Zoom and those of folks who are going to be watching this a month, 2 months when the weather is crappy and they're stuck inside and want to know about chickadees. French and Pickering, I want to say a few words about the organization. We were established in 1967. We've worked with landowners, townships, supporters, the state, the county to purchase and monitor easements over 13,500 acres throughout northern Chester County. One of those special spots you're going to hear a lot about tonight is Templin Woods, which is over 550 acres. With your continued support, we're going to keep working every day to protect the scenic and the cultural landscapes throughout the region, to create opportunities for you, your family, visitors, the public of any nature, any walk, to connect with nature through our trails, the preserves, these environmental educational opportunities, programs, and lectures. Thanks to our wonderful Penny Hunt and the program committee throwing these things together, and the wonderful volunteers like the Worths who turn us all on to birding and really open up a new world of nature to so many of us. And we're so grateful for that, and Audubon and all of our partners, and not to mention our hosts, Historic Yellow Springs, that really generously allows us to have these in person so we can offer that wonderful place to gather and connect around these topics, as well as the virtual experience. So hopefully a little something for everybody. Tonight's sponsor is Patriot Auto Group, and they are really amazing. Jason and Melissa Owens are the owners. They have a couple of different franchises, and they not only support very generously the work that we do at French and Pickering, they also put a stronger easement on land that they purchased in Charlestown Township. So they are members of the community, very conservation minded, and I'd encourage you to kick the tires at one of their dealerships if you're into a newer, yeah, you saw what I did there, yep, a newer or used vehicle. So they are very generous and really supporting conservation in a lot of different ways. And speaking of people who are very generous, with his time this evening, Dr. Robert Bob Curry, he began studying birds during high school in his native Massachusetts, before continuing in ornithology as a college student in New Hampshire and Nova Scotia, where he has family roots. He completed doctoral research at the University of Michigan, investigating social ecology and conservation biology of Darwin's mockingbirds in Galapagos, before carrying out field studies of Florida scrub jays as a postdoctoral fellow, and then as a Villanova faculty member beginning in 1991. In the late 1990s, Bob and many of his undergraduate and master students began studying Pennsylvania chickadees, aiming especially to increase understanding about the causes and consequences of hybridization in this locally abundant and easily studied group of birds. So without any further ado, I will invite Dr. Curry up to begin the presentation.
Thank you for that. If I can navigate a guest computer. Thank you for being here. I'm happy to be here. It's a nice room to talk to you about work we've been doing for now 25 years here. We call the site Great Marsh. That's kind of an extension of the actual marsh. We don't work in the marsh. We work in the hills up above it. So the naming of the preserve as Templin Woods actually better matches. So if it matches the layout of the site. So if we had started now, our acronym for the site would be TW, but I will go back and forth between.
I'm sorry, I'm going to have to interrupt you for a second. They just notified me that someone, if anyone here drove a Toyota RAV4 that's cream colored, it was parked in a fire zone and they will get towed. So sorry, please.
That's important.
Yes.
Okay, so anyway, if I refer to Great GM or Great Marsh, I'm really Templin Woods. So I'm going to talk tonight about this site as a piece of our larger research program investigating hybridization between black-capped chickadees and Carolina chickadees, and I'll explain how Templin Woods fits into that. So that'll be part of the talk. And then the other thing I wanted to do was to highlight some of the work we've done that's not really about hybridization. It's just an opportunity to investigate Carolina chickadees for their own sake. Those are the resident birds at a place like Great Marsh this far south. They're not only Carolina chickadees there, because in some winters we have black-capped chickadees that also arrive and visit from the north, and then they go away again. So those are snowbirds. But because they're part of, they mix into our regular study, we figure we might as well investigate how they fit in also. So this research program that I'm going to be describing, some pieces of it, not the whole thing, has, I certainly in no way did it myself. I did this work with a long list of students, mostly Villanova master students. We don't have a PhD program. So I've relied on the good work of many master students. So the ones I've highlighted here are master students who focused all or part of their work on the Carolina population at Great Marsh. I've got other students that helped more with the work that we did in other parts of the research program and the sites that I'll mention, including Hawk Mountain. So they are important too. They certainly contributed to some of what I'm going to be showing you. And one of the nice things about working at a place like Templin Woods is that it's close enough to Villanova so that it's not too difficult to get undergrads involved as well. The farther you have to travel, the harder it is to get undergrads to stay engaged, and just the logistics of getting them to and from the field sites gets more problematic. We even have one couple that did their masters together at Great Marsh, and then the next thing they did was went off and got married. And they're still married. I don't think they're hybridizing. Okay, so hybridization is a big topic in evolutionary ecology. Lots of people study hybridization in lots of different organisms. It's quite common in nature, distressingly so for bird watchers who like to think that every bird is identifiable on a Christmas count. Sorry, that's not always true. My PhD advisor, Peter Grant, he and Rosemary wrote a paper now about 20 years ago where they estimated that perhaps as many as 10% of all bird species have been involved in hybridization at least occasionally, once or twice. A more recent survey put that number up to about at least 5% of bird species are regularly involved in hybridization somewhere. And then hybridization happens in a lot of other animals. In plants, it's quite common with wind dispersed pollen and things like that. So understanding what hybridization is all about generally has two layers to it. On the one hand, we want to know, why is it happening? Because the whole textbook, high school textbook definition of a species is goes back to Ernst Mayr. It's called the biological species concept, is a group of organisms that can breed with each other and have babies, but not breed with anybody else. That's a species. Well, hybridization is interbreeding between two members of different groups that, yes, they can all breed with each other, but they can also breed with the other group. So it's not the textbook definition is inadequate. This doesn't reflect what really goes on in nature. So understanding the factors that cause individuals of one population, one lineage, to crossbreed, to interbreed with members of another lineage is an interesting thing for two reasons. One is that it has to do with understanding what makes the two entities distinct species in the first place. Because if hybridization happens all the time, you no longer have two distinct species. They just blur together. And there are even some populations. There's a bird, for example, in, I think it's the Italian sparrow. I always get this mixed up. I haven't seen it. The Italian sparrow is a hybrid. The entire population is a hybrid. It actually turned out that one of the birds I worked on in the Galapagos, my main study population was a hybrid population. I had no way of knowing that when I was there, but later people did the phylogenetic work and figured out that that was the case. So understanding what can cause an individual, and in animals, we're usually talking about mate choice, and in most animals, mate choice is driven by the choices that females make in choosing males, not always, but more so than the other way around. So the question becomes, what would cause a female of one species to not mate with a member of her own population, but instead mate with a member of a different population? So that could be things like she's getting confused about the cues that the animals of the two species are sending out, and she's not following the species recognition cues that would normally operate. So that could be something about vocal signals, songs, calls. It could be morphology, size and shape, or it could be their behavior, dance or a display of some sort. Could be all sorts of different things. So that's what we mean by phenotypic cues, cues about the male that the female is responding to. So maybe she's getting confused. He might be confused too. He might not know who he is. And he may be trying to attract a female of the wrong species because he's imprinted on the other species as potential mates as well. And then there could be a more ecological driver, like in some situations, you'll have one bird that's very abundant and another bird that's rare in a geographic area. That's more likely to push the rarer species to engage in hybridization because they have very few mate choices. So just numerically, they get swamped out. That's becoming more of an issue with climate change. As ranges shift, you get populations moving into new areas, and initially there can be numerical disparity between the previous residents and the new arrivals that can result in hybridization, as well as geographic ranges overlapping where they never used to overlap before. So it's timely to understand some of this stuff. And then that takes it, once it's happening, then we also tend to be interested in, is it a good thing or a bad thing? Well, usually it's a bad thing. There are cases where the hybrid individuals are strong and vigorous like a mule, but of course mules are sterile. So in terms of their evolutionary fitness, it's a bad thing, even if the animal itself is healthy and doing well. But sometimes the hybrid animals are not healthy, and that can cause them to have poor success, even if they're not infertile. They may be able to have kids, but they may not have as many kids. So we talk about fitness components. That's what are the things that influence the reproductive success of individuals of mixed ancestry compared to individuals of unmixed ancestry. Just as an aside, I try to avoid, and I try to get my students to avoid using the terminology about pure species, because frankly, that's pretty racist. So I use the term unadmixed to refer to a population where everybody is from the same lineage. And if it's a hybrid population, that's an admixed population. So it's turning the mixture of the genes. Okay, so there are lots of different ways to study hybridization. You don't have to do what I do. You can, so museum-oriented biologists go out and collect specimens and then study them in the lab, either by studying their morphology and their plumage and things like that from museum specimens. And these days you can get a lot of DNA from the museum specimens and study that. But if you really want to fully understand what's going on with hybridization, with both the causes and the consequences in animals, you really have to study behavior. And I prefer, rather than observing behavior, then shoot the bird and find out what it was that I was looking at, I'd rather study the bird and let it go about its merry way and continue to study it. So long-term study is what I was trained to do. And it allows us to address a lot of the fundamental questions about hybridization. Now, the challenge with purely observational study, even if it's long-term, is that there are limitations to your interpretation of the results. You could get fooled by a correlation between two factors that actually is not cause and effect. That's a standard thing we all teach our students, that correlation does not equal causation. So sometimes you have to do experimentation to test things in a more rigorous way. So we've done some of that as well. That's not really my background. My background is more long-term studies. I'm trying to think of Peter and Rosemary Grant working in the Galapagos for more than 50 years. How many experiments did they do? About 3. And they published many, many papers about Darwin's finches. And they didn't have to do experiments. They're looking at measuring patterns and analyzing the patterns between islands, within islands, all that stuff. So that's the heritage I come from, the intellectual heritage. Well, in the mid to late 90s, I had been working as it was mentioned in the introduction that I was working in Florida on Florida scrubjays, which are wonderful birds. I love studying them, but it's a long way away. So it was hard for me and hard for students to work down there. So I started looking for a system closer to home that would be tractable using the kinds of methods that I wanted to use. And I gravitated to the chickadees actually by walking around at the marsh, helping a master's student who was studying water snakes. But as he was working on water snakes, I got to know the property and kept hearing chickadees and thought, hey, I could work on chickadees here. And we already knew that this hybridization was happening in our eastern chickadees. So this is the range map with the blackcaps up in the north, the Carolinas in the south, and we are right here on this red swath, which is called a contact zone or a hybrid zone, where the two species overlap. It's called a parapatric range, where they just barely overlap. They don't completely overlap like a yellow warbler and a common yellowthroat. Their ranges overlap over thousands of miles, but they don't hybridize. So they figure it out and they coexist over a big geographic swath. You're actually more likely to hybridization in this kind of situation where only a small percentage of each of the two species ever sees the other species. So they get confused. So this is our eastern hybrid zone, and this was known that this was happening for quite a while. People had written papers back in the 1930s about hybridization between these two species. So when I made up my mind to start studying chickadees, I knew that hybridization was one of the things that we would probably want to investigate. And especially given that from here, as the chickadee flies or the crow flies, to get up to the area where it's black-capped chickadees is only 30 or 40 miles away, we thought. It's running away from us, so you have to go farther and farther and farther to get up to where the black-capped chickadees are. But that came later. Okay, well, just a little bit of technical background about the two species. So these are our two species, Carolina chickadee and black-capped chickadee. This is a phylogenetic tree, an evolutionary tree, and the numbers just measure the branching, the similarity between the two species. But what you can see here is the black-capped Carolina chickadee and black-capped chickadee are each other's closest relative as species. So presumably, we don't know for sure when they split, but it's quite a while ago. They are more different from each other than a bonobo is from a common chimpanzee. They are about as different as you are from a bonobo in terms of percent DNA. But you don't look like a bonobo, and you probably wouldn't hybridize with one. They're doing it. Okay, so it goes back a ways. But the fact that they're each other's closest relative does matter because it means that they're pretty similar. So the thinking is that sometime back in the Pleistocene and the ice ages, the black-capped chickadees got shoved over into areas on the East Coast that were not covered with ice and got isolated there. And meanwhile, the Carolina chickadees got shoved way down into Texas and Mexico. And there was a period of time for hundreds of thousands or a million years where they were geographically separated and diverged. And then when the ice retreated and the blackcaps moved explosively from east to west all the way to Alaska, and meanwhile the Carolinas slid northward and then they banged back into each other. So that's the background. Now these other species are relevant too. I'll say a little bit along the way about mountain chickadee. That's the next most closely related bird in the group, and colleagues of mine, collaborators of mine, are now studying mountain chickadee and black-capped chickadee, where they hybridize out in the Rocky Mountains. It's a different thing because out there the pattern is the blackcaps are down in the low elevations and the mountain chickadees are in the tops of the mountains. So the hybrid zone is every mountain has a hybrid zone. So it's a mosaic of little isolated island-like populations. So there could be different things happening in different mountain ranges all across the Rockies. So it's a really interesting situation, but much more complicated than what we're dealing with here. We just have this north-south split and this east to west seam between the two species. It's all these other guys down here are the brown-capped chickadees. So Hudsonicus is boreal chickadee, rufescens is chestnut-backed chickadee. They hybridize with each other rarely, but only in like northern British Columbia. They don't overlap, but they can hybridize. They virtually never hybridize with the black-capped species. So even though boreal chickadee and black-capped chickadee overlap across almost all of Canada, they virtually never hybridize. There's like 2 records of hybridization ever between those two species. What's different about them? Well, these brown cap guys don't sing. The males don't have a whistled song. So the black capped ones all have the same signaling system and just different variations on it. Whereas the brown cap chickadees do different things. They just behave differently. They have calls, but they rely on calls rather than songs. So that kind of explains why this is a very big difference between these brown capped birds down here. Cinctus is the gray-headed chickadee way up in Alaska and over into Siberia. So you got the black cap chickadees who can hybridize. This top one is the Mexican chickadee that we don't know very much about. It's a really strange one. So the three I've shown here, Mountain, Carolina, and black cap, are the ones that get the most attention. So increasing, you're going to hear more about black caps and mountain chickadees in coming years from the very good people that study them in Colorado. Okay, well, we're not in Colorado, we're here. So this is the southeastern corner of Pennsylvania, and these are our 4 study sites that we set up. With the Carolina, this is the acronym for Carolina chickadee, CACH. That's the bird banding lab acronym. So we gravitate to that. Every bird around here has a 4-letter code, if you didn't already know. So their range is, this is essentially Chester County and pieces of Lancaster County, and maybe a little bit of, well, now most of Montgomery County and Bucks. It's all Carolinas. So when we started, Great Marsh was already all Carolinas. There had been a study back in the 1960s in the area near Great Marsh, and there was some evidence of some blackcap chickadee behavior in the area, some songs that were more like blackcap, but that was 30 years before I started. But we think that the hybrid zone has been sliding northward for quite a long time. But what the entire duration of our study, Great Marsh has always been Carolina chickadee, except for these visiting blackcaps, but the resident birds are Carolinas. The place where we first started zeroing in on hybridization was Nolde Forest up near Reading State Park. It was a wonderful site to work at. When we first started working there, everybody was singing blackcap song, and I thought they were all blackcaps. We did the genetics. Not one of them was a blackcap. They were all hybrids. So we got fooled, but that meant that we were right in the middle of it. So that was good. So we did a lot of work in the first decade or so of the research program at Nolde. And then we realized, ooh, these aren't blackcaps. We need blackcaps. So we went up to Hawk Mountain. We got blackcaps. And now we have something other than blackcaps because the hybrids in Carolinas have taken over there too. So then we had to add Tuscarora way up here. Back in the about 2006, we started working at Tuscarora to get more unedmixed blackcaps. And now we have hybrids there too. So they keep running away from us. And I stopped at that point because I didn't want to go to Scranton, too far away. Okay. So at all those 4 sites, so our approach is, it's not really a transect. If it was a transect the way evolutionary biologists who collect specimens do transect, I would have had 20 locations from north to south or east to west or whatever. So in the museums, you see specimens from a transect like that. So we're using these 4 populations to get intensive data from each of those spots, but still on a north-south line. So at each of the locations, we rely not on chickadee natural nesting behavior, because I'd have a sample size of about 10 after 25 years. It's very hard to find active chickadee nests. We've gotten pretty good at it, but we would prefer that they don't nest in their natural situation, because their normal nest is in a dead tree about 3 or 4 inches in diameter, never an oak. It's almost always a birch, a poplar, sometimes a really old maple. They actually don't like sweet birch. I don't really understand why not, but they like paper birch and things like that. So they nest in it right before the tree falls down, because they like it when the wood inside is really rotten, which means if you find the nest and it's 12 feet up, it's going to fall over if you try to climb the tree to get at the nest. But we do find some. The lowest nest I ever found was about ankle high. The birds were scolding me, and I was looking for, where's the nest? And I looked down and realized that right by my knee was a snag, and the birds were going into a hole about 6 inches off the ground. And that's good, because when it's that accessible, I can cut the front off the wood and get at the nestlings to band them and do what we do, and then close it back up again and use that wonderful thing, duct tape, to close up the snag, and off we go. And then by the next year, that snag will have rotten and fallen down. But we don't rely on those. We rely on these things. So if you walk around in Templin Woods, you'll see about 100 of these plastic tubes with a hole on the top. That's for the chickadees, and there's a floor right here. So those are our artificial snags. We didn't invent them. Tom Grubb and his researchers out in Ohio that were studying chickadees there figured out that this was a good way. You can use a regular nest box too, but bears like nest boxes because if you're attached to a tree, a bear can get at it, and they just smash them. So these things are easy to just stick up, jam a post in the ground and put it up. We can move them around. They're pretty easy to maintain. It's sewer pipe. We just go to Lowe's and buy sewer pipe. So actually pretty cheap too. Once we have an active nest and we can count the eggs, we pop the top off. We can just look inside and count the eggs. We take note of the eggs. All of these eggs look the same. They were all laid by the same female. That's not always true. They don't get parasitized by cowbirds, but they do get parasitized by other chickadees under special circumstances. So I can answer questions about that later if you're interested. Then they hatch and they get sort of cute. Initially, they're pretty ugly. Then right before they fledge, they're really cute. And then we also are able to put a mist net up in front of the active nest and get the parents as they're coming and going with food for the kids. So that's how we catch most of the adults. We also, and then we get blood samples because not so much at Templin Woods, but at all of our other sites, we don't know what we're looking at. So we have to, the only way we can figure out who the bird is by analyzing its genetics. And so far, Ancestry DNA doesn't have a test where I can do a cheek swab on the bird and send it to them. We have to do it ourselves. They can do your dog, but they can't do your bird. They need to work on that because they do 700,000 SNPs. I'm going to talk about 10 in a minute. Okay, so this is Templin Woods. We don't cover the entire property because we don't like to walk any farther than we have to. So this area here is too far away from the roads. It's just too hard. So we work in off the road here, and Templin Road is right here. And this is the parking lot for the start of the trail. So the trail now is a loop like this. And this, so we have 3 sections. That's not ideal. It just worked out that way, that logistically, that's what we could manage. So each of these dots is one of those plastic tubes. So they're at least 100 meters apart. We can get, so there's 100 at Templin now. In a good year, I'll show you the numbers in a minute, we get up to almost 50% of the snags have a chickadee nest in it. So they do like them. And that means we don't have to go searching for the nest. We know where they are. So we just have to go around and check 100 snags instead of searching endlessly for natural nests. The challenge, though, is we also simultaneously have to do the same thing at Nolde, Hawk Mountain, and Tuscarora. And we have 200 snags at Hawk Mountain because we're trying to get a larger sample there because it's where the hybrids are, and it's on a mountain. So doing the same work, we love Templin Woods because it's so easy to move around compared to Hawk Mountain. I've almost killed myself a bunch of times at Hawk Mountain because it's really rugged. So when we catch the birds, we've got all these different band colors. We either put a metal numbered band, a USGS band, geological survey, and then 3 color bands on, or I'll talk a little bit later. We also use electronic tags called a pit tag that takes the place of these 2 bands. It's a little bigger and it has an electronic tag embedded in the band. So then each bird, ideally each bird has a unique combination. So this bird is silver blue, yellow, hot pink. And so to identify the bird, you need to see all the colors. And these are tiny little birds flitting around up in the treetops. So it's a challenge. Often we have to put the net up and catch them to know who we've got. The nestlings, we don't put the color bands on because most of the nestlings we never see again. But if we come back the next year and we have a bird who's now a breeder at a snag who only has a silver band, that's one of ours. That's one of our nestlings who has fledged and probably dispersed a little bit. So we actually have a really good data set on chickadee dispersal and how far they move from home. Okay. So it's a good system. I said they're easy to study. It's not perfect. This is an example of one of our feeders with electronic system attached, and the squirrels have opened it up and eaten all the seeds and destroyed it. And they also like to chew the wires. And with these electronic systems for reading the tags, if the wire gets broken once, it's dead. So then we have to go back out and replace the antenna and start over again. So gray squirrels are not our friend. Black bears are not our friend. At Templin, it's not a problem, but at Hawk Mountain and at Tuscarora, the black bears love these things. They're crunchy on the outside and chewy on the inside. So they'll eat a whole brood of nestlings, or they'll try to trap the incubating female. And then we get these guys. Whoops. See if the video works. Video doesn't want to work. Come on. Darn. Well, what I was going to show you is that's a nest cavity with the antenna around the entrance, so we can measure the parental behavior of the male and the female. The video shows a house wren going in and not removing the eggs, which they do routinely, or the nestlings, which they sometimes do. The house wren actually hauled the incubating female right out of the tube. And we've had chickadees get scalped by the wrens. The wrens are vicious, so we don't like wrens. So they're actually, that limits our sample sizes, because the wrens will destroy the chickadee nests, and if they go off and nest somewhere else where we don't find the nest soon enough, we lose that data point. Okay, but it's, those are our three. So the bears just swat the things and knock them over, and then we have to put it back up again. But that's not too bad, unless the bear gets really good at it, which sometimes happens. But the wrens are really tough, and they're very abundant around here. So in some years we lose a lot of chickadee nests to wrens. Okay, so for this hybridization, so the first part of the talk is all about hybridization, and then I'm going to come back to talk about some of the Carolina specific stuff. For the hybridization work, as I said, we need to be able to tell the two species apart, either directly or we go to the genetics. The direct part is manageable sometimes. So this is a case, I've got these two birds in my hand simultaneously. I caught them in the same net. This was here at Templin in the winter. So this was a resident Carolina chickadee and a visiting black-cap chickadee from somewhere far away up north. So it's a very big black-cap chickadee. So the things that stand out is the brighter white in the shoulder and the edge of the secondaries. So along here, it makes a hockey stick shape, a white L. They also have a longer tail. So that tail is proportionally a lot longer. So Carolina chickadee tail measures about 53 millimeters, and a black-cap chickadee tail can be 69, 70. So it's like a 20% difference. So with practice, you notice the difference in proportions, that it really seems like a long tail on the blackcaps. But this is a case where these irruption birds, these visiting blackcaps, come from way up north. We don't know exactly how far away they're coming from, but we know that at banding stations in places like Braddock Bay on Lake Ontario, in some winters or fall, they'll get flights of thousands of chickadees, and those chickadees keep going. So they're coming down from Ontario, upstate New York, Maine, and working their way down, all the way down into Maryland. Sometimes they'll go all the way down to Virginia, and we get them here. So in an eruption year here, you could have, for every 10 resident Carolina chickadees, you could have 5 or 6 blackcaps mixed in. They don't stay all winter. They just come for a few months and then they leave. Okay, so if you get them, those extreme differences, then you can tell them apart. Even on a Christopher count, I could tell them apart. But the hybrids are exactly halfway in between. So good, and there's everything, it's a gradation. So a lot of the birds, we just can't tell. So I've gotten pretty good at saying, okay, that bird's a hybrid. So to me, it jumps out as not representing either of the two extremes. So if you go to Hawk Mountain and go to the visitor center and look out the windows at the bird feeders now, virtually every bird at those feeders is a hybrid. They're all intermediate in size and shape and plumage. Now the songs are very distinct. So I've gotten pretty good at imitating the songs usually. So rather than playing it on the computer, this is a blackcap song. This is a sonogram. So this is time and pitch. So it's a sonogram or spectrogram. And the blackcap song is a very simple cheeseburger, which you would never hear here. You have to go up to Hawk Mountain to hear it. Even at Nolde, they don't do this anymore. But it's just those two notes, and it's the same almost everywhere. So from Massachusetts up to Maine, Nova Scotia, all the way across to Alaska, every blackcap chickadee sounds the same. And that's the only song they sing. The only places where they sing something weird are isolated pockets like Martha's Vineyard. There's strange things go on in Martha's Vineyard. So they mix it up a little bit there. But in general, it's very simple. Carolina songs are much more complicated. So their basic song here is this high, low, high, low. It's much higher pitch, so it's harder to imitate. High. So you usually hear the second note and the fourth note. I'm getting hard of hearing. I'm having trouble hearing the first and third note. So here it's always high, low, 4 notes. Sometimes it's 6, high, low, high, low, high, low. But they have a second song too. So each male Carolina chickadee can sing more than one song. So that complicates things. And the problem with the songs is they learn them. So if they were next door, if a Carolina chickadee male grows up next door to a blackcap male and his father dies, the only male he hears is a blackcap, they're going to learn the blackcap song. Okay. The calls are only a little bit better. So the most basic call is their chickadee call, their eponymous chickadee call. So it's got letters that go with it. We'll come back to that later, but it's A, B, C, D, 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 D. And the note you really hear is the D note. So I can tell these apart after 25 years. If the bird goes chickadee, D, 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 that's a blackcap. If it goes chickadee, D, 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 that's a Carolina. If it goes chickadee, D, D, it's a hybrid. You got that? So the point is the D notes of the hybrids are quantitatively intermediate. They're shorter. They're halfway in between because these notes are shorter than those notes. And these notes are higher pitched than those notes. And the hybrids calls are halfway in between on both. And the calls are not learned. So we think that the calls pretty accurately reflect their genotype. That's one of the last things we're working on in my lab is nailing that down. So I'll come back to that. Okay, so we got started on all of this, as I said, in the mid-90s. I'd been shopping around for a system, and we started. So the first thing I had to do in 1997 was figure out, where do I go? So I spent a lot of time, got to know all the Nature Conservancy properties around here, Natural Lands Trust, went everywhere. French Creek State Park, because I didn't know where the hybrids were. I was trying to find the hybrids. I got totally fooled by Nolde, but it turned out that Nolde was exactly where I needed to be. And then we added Hawk Mountain, which was fine because it's a protected area and they were welcoming of us. So we proceeded with that. And then we had to check about, are the birds who we think they are? So we did what's called a structure analysis. This is a standard way of representing genetic differences across individuals that might be in different populations. Okay, so what you do, each vertical bar on this graph is one individual. Okay, so over here, every individual, so there's about 30 individuals there, they're all almost entirely a black bar. The ones over here on the far left, those are white bars. So the genetic markers we used for this told us that the birds at Great Marsh were almost entirely unadmixed Carolina chickadees because the markers we're using were completely different for Carolina versus blackcap. And you'll see at Nolde, a lot of the bars are gray. Those are the birds that are both, genetically both. So they got one chromosome from mom, one chromosome from dad. The chromosomes don't match. So those are birds that are hybrids. So this was back at the early part of our study, the first 5 years. You can see that Nolde was the thick of things. So Nolde was a very mixed up population. So this was the data set that tipped us off. You can also see that at Hawk Mountain, there are a couple birds with black bars. So even back at the very beginning of our work at Hawk Mountain, we had birds that weren't unadmixed Carolinas. We had a couple hybrids starting to move in. Meanwhile, at Great Marsh, maybe one or two of these birds had leftover blackcap genes from 30 years ago, which would have been their great-great-great-great grandfathers or something like that. That's possible. because we knew that there were some blackcaps in the general area. Okay, I'd be happy to answer any questions if anyone has them. This is the most technically tricky part of the whole talk, is to talk about how we do things now. The markers we used for that first analysis were okay, but hard. They were expensive and time consuming and tricky to get clear results. These days, what most people do, including Ancestry DNA, to characterize a genotype, if you don't want to do the full sequence where you get every ACGT, which you can do if you've got a few $1,000s, but if you're trying to do individuals, you want to keep the cost to like $10. What you do are called SNPs. So Ancestry does 700,000 SNPs when you send off your spit to them. It's amazing how much detail they get. We do 10. So it's cruder, but it's still the same idea. So what we want are base pairs, either an A, C, G, or a T, where black-capped chickadees have an A and Carolina chickadees have a T. So it's a diagnostic difference in one base pair. So what we do is amplify the DNA using PCR, which is a method for finding a section of DNA and making a whole bunch of copies of it. So you end up with just that chunk of DNA magnified about 1 million times. So now you got enough DNA from one little drop of blood to work with. And then we throw in an enzyme that's going to try to cut it. So we have 10 of these. So the enzyme is looking for, is that base pair at that one particular spot plus the adjacent ones? It's looking for a sequence like AAT. If it sees AAT, it cuts it. If it sees AAG, it doesn't cut it. So that's the diagnostic difference. That's a SNP. So we're getting, if it cuts it, now we got 2 small pieces of DNA. If it doesn't cut it, we've got 1 big piece of DNA. And that's what we measure in the lab. So you're looking at fragments. And this is pretty cheap. Undergraduates can do this these days, really. Not perfectly, but they can try. Okay. So one of the biggest advances in the whole research program was in the early 2000s, in 2010 or so. Scott Taylor, who was a postdoc at Cornell, got in touch with me, wanted to explore the possibility that we had enough data to investigate climate change. So the plan was to compare the genetic composition of the population from 2002. This is essentially the same graph I showed you before, just turned on its side. So we got the Carolina chickadees at Great Marsh, the mixed population at Nolde, and then the mostly black-cap population at Hawk Mountain as of 2002. So this was based on about 1,000 SNPs. So each SNP has a location somewhere between Great Marsh or Villanova and Tuscarora, where it switches from Carolina genotypes in the south to black-cap genotypes in the north. So that's what this changeover is, the change in likelihood of the gene being a black-cap gene as we go from south to north. Okay, so that's called a climb. You don't need to worry about that really. What matters is if you look at the pattern in 2002, so we've got the beginning of hybridization at Hawk Mountain. If we come back a decade later, there's a lot more gray bars and a lot more 50-50 bars. So the Carolinas and hybrids moved into Hawk Mountain in the 10 years. Nolde, we lost all the hybrids, except for 1 or 2 slightly admixed birds. So those are birds who had, of their 8 great-grandparents, 1 was a black-cap, and the other 7 were all Carolinas. And then at Great Marsh, it's all Carolina. And Tuscarora, when we did this, was all black-cap. So the point was that things had shifted in the direction of more Carolina genes at both Nolde and at Hawk Mountain in 10 years. And we did the math, and it turned out that that change corresponded to a northward shift in the position of the hybrid zone of about 1 kilometer a year. So the whole zone, this geographic shift in where it changes over from Carolina black-cap, moved north in a decade 10 kilometers. So that's 1 kilometer a year, and it's continued. So in the 25 years I've been doing this, it's moved 25 kilometers. Well, now we're getting, so that explains why Nolde used to be hybrid and now it's all Carolina. And Hawk Mountain used to be mostly blackcap and now it's all hybrids, and even Tuscarora is getting the hybrids. So that's how we do the work. And this is another way of looking at this is the proportion of Carolina genes. So at the beginning of the study, it was mostly blackcaps with a few hybrids up here, and that trend has gone up and up and up. So you can see the Hawk Mountain population as of 2020, 2021, 2022 is very mixed up. We've got everything from, we've got actually no unadmixed blackcaps left anymore. These are all hybrids. So virtually everybody at Hawk Mountain right now is a hybrid. Meanwhile, back at Templin, everybody's still a Carolina. Yeah.
So are you convinced that the extension of the range is due to climate change and not any other?
So that was the question we addressed in the paper where we presented the genetic results, and the collaborators I worked with at Cornell used eBird data to map out the position of the only Carolina, only blackcap, both over a much broader geographic area. And then they correlated with climate data. And what they found was the position of the hybrid zone in 2002 corresponded with a particular winter temperature. And the location of that winter temperature in 2012 had shifted 10 kilometers farther north. So the correspondence between the degree of change in the coldness of winter nights matched exactly where the hybrid zone had moved. So it's still correlational, but it's pretty convincing, and the pattern has held up. Okay, so I'm going to switch over more to talking about consequences, because there are some. What happens when they hybridize is initially it's really bad. This is a typical Hawk Mountain nest. 8 eggs, 1 nestling. So the other 7 eggs had never hatched. They either weren't fertilized in the first place because the sperm and the egg were of 2 different species and they didn't get along, or the sperm fertilized the egg and the embryo immediately died and never hatched. And we think it's a mix of both things going on. But you can see that the hatching success, as I just showed you, the proportion of hybrids has gone up and up and up. And meanwhile, the hatching success has gone down and down and down. This is at Hawk Mountain. So that's really bad for consequences, because your fitness is dropped by 80% right off the bat. Because if your reproduction goes from 7 kids down to 1 kid, that's a huge deficit. So anybody around you who's avoiding hybridizing and producing 7 kids is outnumbering you big time in terms of children, grandchildren, great-grandchildren. Darwin was right. So that's just natural selection favoring the tendency to not hybridize. Okay, but we do have data from the other sites. So at Great Marsh, no change in hatching success. We had no change in the genetics, so we wouldn't have expected any change in the hatching success, and that's nearly a flat line. We do have some nests that have less than 100% hatching success, but that's for a variety of reasons. But most of the nests are way up around 95, almost 100%. Cavity nesting birds generally have very high hatching success. This is Tuscarora, where as of 2018, we still didn't have very many hybrids, so it's still doing really well. Now it's started to go down. And at Nolde, it actually went up because we got rid of the hybrids. The hybrids got shoved out by the Carolinas. So as Nolde became more and more dominated by Carolinas, the hatching success improved. So what's going to happen 20 years from now, and I won't be studying it, is the population in Hawk Mountain will shift from being a hybrid-dominated population back to a Carolina-dominated population. It'll repeat the history we saw at Nolde and the
hybrid group, do they just die out, or do they become taken over by the incoming group?
Both. I mean, eventually they die out. They just don't produce enough mixed children to maintain. And meanwhile, their descendants may be breeding with unadmixed blackcaps farther north and producing new hybrids. So you get this wave of hybrids where it's bad to be a hybrid, but they keep doing it. And so it keeps moving up into the blackcap area. that's the part that in the grand scheme of things is very hard to study that broader pattern. So we're just getting glimpses of it.
Question.
Yep.
On that, I'm trying to reserve questions, but yeah, but that helped me. I couldn't help but think about this. So basically, the hybridization is a continuous effect.
Yep.
But spatially, it's temporal.
Right. That's right.
If it was to be static, let's say climate was not a factor, there's all these things.
Yep.
Would you suspect that that would then create a zone of inhibition, a true speciation between them, given enough time?
The prediction would be that what would happen is called reinforcement, where the unadmixed birds who prefer a mate who's of their own species would have much higher fitness than the hybrids, and they would get it right. They would figure it out. But that would only work. So it's where climate change is kicking in as a bad thing is creating this dynamic where it never has time to resolve itself. So they can't figure it out. Yeah. It's happening too fast. So they're getting naive birds coming in contact with hybrids and they just perpetuate the error. That's our interpretation. Yeah. So these are the data from Nolde just to show that the Carolina-ness of the population has gone up and up and up. Okay, and this is the hatching success pattern at Hawk Mountain. We've got the average hatching success at Hawk Mountain now is like 40% or less. So we do have a few pairs that do quite well at Hawk Mountain, but now we think that some of those pairs are actually Carolina pairs because they do fine. So there may be one or two leftover black cap pairs, but it's mostly new Carolina pairs that are going to pump out a whole lot of kids, and those kids are going to dominate the population next. And the hybrids are not producing very many kids. So this is just the graph showing you that nestling production follows immediately from a number of eggs that hatch. Not much else happens after. They don't die of starvation very often. So if the eggs hatch, they generally fledge. Yeah.
I have a related question. You're looking at this chickadees in the aggregates. Say you're looking at rebirthing.
Yeah.
You would see this dip in total population
Yes.
moving north.
Yes, that's what we have right now at Hawk Mountain. Our sample size at Hawk Mountain the last few years has been very low. Just not very many of our tubes have chickadees nesting in them because we got this trough caused by the poor production of the hybrids. We're seeing it right now at Hawk Mountain. At Nolde, we went through that at the beginning of the study, and now Nolde's chock-a-block full of chickadees because they're not hybrids anymore, and they're everywhere. Yeah, that's spot on. Yeah. Okay, so the take-home messages from all of this is that the Carolinas are invading the southern edge of the blackcap range. The blackcaps are also moving north at the northern edge of their range, so the whole species is shifting north. Hybridization is not quite finished at Hawk Mountain. It's happening pretty fast. It is almost completely finished at Nolde. We've got a few residual leftover blackcap genes, but we do expect that the Carolinas are eventually, assuming the climate continues to warm up, that the Carolinas are going to win.
You said a word, fast. Will you be able to show that it's happening faster than it did before?
I won't. Well, actually, if anything, it's likely to slow down because now we're in the mountains. So as you go up in elevation, it gets cooler. So that's going to moderate. Everything that's happened up until recently happened in the lower elevation areas where just the winter temperature warmed up very quickly. But that's not going to happen in the Poconos as fast. But surprisingly, the hybrid zone out in the Midwest is not moving very fast northward. But the pattern of warming is not the same either. So warming is regionally variable. But around here, it's warming winter, overnight low temperature, which is what matters to a small bird physiologically, has gotten a lot warmer. It's very rare to get a night with 10 below anymore. Well, that's part of it. Yeah, the oceans are getting warmer, so there's a lot of heat coming in off the ocean. Yeah. But in Ohio and farther west, the pace of northward movement is not nearly as fast. But those are good questions. Okay, so I want to shift gears. I'm happy to answer more questions about hybridization at the end, but I wanted to touch on, just give you a flavor of what we can do with just Carolina chickadees for their own sake. I'm I still am surprised that we are really the only operation anywhere that studies a large population of Carolina chickadees with individually marked birds. I don't get it. They're easy to study. They're cheap. It's manageable, and nobody else does it. So you think of all the universities and colleges in the Southeast, everybody could be studying Carolina chickadees. They're everywhere, and nobody does. There was a guy at Tennessee for a while, but he retired and stopped doing his work. So it's left us in the position of learning more about Carolina chickadees for their own sake than really anybody else. So as I go into retirement next week, next Friday, one of my jobs is to go back through our older data and get more of this basic information published, because it's been slow to get that done. We've been focusing on the hybridization stuff, but some of this Carolina stuff deserves to get published. And also what goes along with that is that we are the only operation anywhere that I know of where we've already got the local birds color banded. So when the eruption birds come in, we know who's who, and then we catch and color band the eruption birds, and we study their interaction with the resident Carolinas. Nobody else does that either. So people get a lot of data about eruption blackcaps at banding stations as they move through, but nobody actually goes out and chases them around in the woods like we do. Okay. So by basic biology, I mean a lot of things. We got ecology about clutch size and nesting behavior, parental effort. We can put the antenna on the nest. I showed you the picture of that with the house wren, and measure how many times the female goes in and out of the nest. We can study their songs, survival of the individuals, the persistence of pairs. Shorter answer is they never divorce. So they stay together until one of them dies, and then they get a new mate, but they can stay together. I think the longest pair we ever had at Templin was 6 years. So to get them both to survive for 6 years was astounding. The average lifespan is about 9 months. Yeah. But the year-to-year survival is about 50%. So of adults. So the chance of a bird surviving 6 years is 50% times 50%. It's 6%, something like that, or 3% to do the math. Good question. Bad weather in the winter. So if you get an ice storm, that's fatal for chickadees because it covers up all their food in ice. They have to eat like once every couple minutes every day, all through the daylight hours, just to stay alive. Hawks, sharp-shinned hawk, things like that. Our incubating females get depredated by flying squirrels. Flying squirrels are quite vicious. And snakes. So there's a variety of predators. Yeah. Okay, so these are, I'm going to give you some examples of the different things that we study. Here's an example of our ecological data set. This is the number of nests each year from the very beginning. So the number of nest snags was not constant. So at the beginning, we only had maybe 25 snags, and we got nine nests the first year, then 15 the next year. Then we kind of stabilized around 2005 or 6 at 100. So the variation since then is real variation. 100 snags out there, and we had as many as 46 one year. So almost 50% of the snags had a chickadee nest in it. And then a few years ago, we had a huge dip. Everybody did. There was a big drop in chickadee abundance regionally, and we don't understand what it was. We know that they were susceptible to West Nile when West Nile first came, which was back here, and we think it might have been a resurgence of West Nile, but we're not certain about that. They do have malaria. We've got samples. They have a lot of avian malaria, but that's not deadly to them as far as we know. So that's just how many nests were active. Then we can do things like, this is basic stuff in ornithology. What happens to clutch size depending on when the nest was started? So the nests that are laid early, I have a nest behind my house where the first egg was laid on April 14th this year, 9 eggs. That's pretty typical for an early nest. If you have a nest laid today, it's getting pretty late in the season, it would only have 5 or 6 eggs. So it goes down. And that's not unique to chickadees. That's a lot of species do that. But it's, this is a really good data set. We got 600 nests. So that's a lot of information just to reinforce that generalization. So what goes along with that is even just looking at the Templin data, we can ask, has the laying date changed? Because if the weather gets warmer earlier, the birds might start laying earlier, and they might follow this same trajectory where they'll lay more eggs. Because earlier is better in general. So if the weather allows them to breed earlier, then we'd expect they're going to produce more eggs. We get even more Carolina chickadees. So that might be part of what's going on. However, there is no big detectable change in the breeding happening earlier, even though winter weather has gotten warmer. Winter weather might not matter to their nesting timing. It may be spring weather, and the spring weather is much more variable. So if you got a cold, wet spring, they're late. If you got a warm, if it's really warm in the end of March, they nest earlier.
Colleague, who's at, what's he at, Villanova University, just co-published a paper on variation in spring temperatures,
Mhm.
and there is a longitudinal.
Yeah.
As well, so.
Yeah, I don't have weather data for Templin, so I should go look and see if I can find some. Yeah, yeah.
But I,
Yeah, I'd expect if I had a big enough data set, I would expect that eventually we will see that. They're non-migratory birds, so they are here and they're affected by the spring weather. Unlike a long distance migrant, they're just going to show up based on day length. And
I think the take home with that paper is that the intense, the most intense variability in spring is getting later. As you go
Oh, yeah.
north,
Okay.
it's later where you get that
Yeah.
high variability.
Yeah.
But,
I mentioned this before. I won't go spend a lot of time on this, but this is just about the song variation that a lot of the generalization I made about the Carolinas having more mixed up songs comes from our work at Great Marsh, including a project that Jim funded, an undergraduate study. Thank you, Jim. It is relevant for the hybrid zone because what our data from Hawk Mountain and Oldie show is that some birds become bilingual. They can sing the blackcap song and the Carolina song pretty well. So it just sounds like normal songs, but they sing both of them. So which are they? Well, the female that might be a tip off. Hey, this guy's a hybrid, but it doesn't seem to stop them from mating with him. And then what happens over time is that as the Carolinas move in and there's more and more Carolina song models around, then you stop hearing the birds singing blackcap song plus the A song of Carolina. They kind of swap out the blackcap song for the B song of Carolina. So having two songs is a Carolina feature, but the hybrids don't plug in the second Carolina song, they plug in the blackcap song. So there's still some interesting details about the song learning that we're trying to wrap our heads around. And now my colleagues, like Scott, I mentioned Scott Taylor, he's a genomics guy. So he can get in there and look at the DNA in the brain, and not just the DNA, but the RNA, which genes are turned on in the brain. He can look at brain regions like the ones that control song learning and look at what's different between a blackcap and a Carolina. So I'm going to be handing off 12,000 blood samples to Scott Taylor and say, have fun with those. Yeah, but that's the kind of thing that he can do now. So they're doing a lot now with cognition, but they could potentially do stuff with songs too. This is an example where we do some experimentation, because to really understand the song, you don't want to, well, one thing, you don't want to be out there at 4 o'clock in the morning with a microphone and then the bird decides not to sing and you wasted a morning. I can tell you from experience that happens. We did get smart about using automated recorders that record, and they're out there right now at Hawk Mountain. But we also did experimentation to try to elicit song, because the bird might know two songs, but it just chooses not to let you know. So you have to force them to do it. So we put two speakers out, and it alternates between blackcap over here, Carolina over there. It goes back and forth, and then we measure where does the bird go and what does it sing in response to the two parent songs. And we've done that at Templin also with the two versions of the Carolina song to see, do they care about the A song versus the B song? So we use the same paradigm for both. So the way we do this is my videos probably aren't going to work, unfortunately, but this is what it looks like. The thing in the middle is the speaker with a model of a chickadee on it, 3D printed model, and then the bird comes swooping in. So if the video is working, you'd see this bird got really upset about that particular song. Meanwhile, there's another speaker 30 meters away where later he would hear the other song. So we're measuring the difference in their aggressiveness to one stimulus versus the other. So I won't bore you with the data, but that's just how we study this phenomenon.
You're collecting those from MacArthur style, like you're out there watching, or is this video derived?
Both. We
Okay.
have the video running, but we're also there with the binoculars, yeah, and also recording the vocal response. Yeah. So we've had a couple projects like that, and this is another one about behavior. This one's a little different. There's a subject within behavioral ecology about personality research, and personality in non-human animals means what you think it does. It means animals have different personalities, just the way people do. Well, what does that mean? Well, it means one person is outgoing and another person is introspective. Things like that. One person's aggressive and one person's passive. Animals do the same thing. So it's behavior that varies from one individual to the next, but each individual is consistent in their behavior. So you can put them into bins. So we've been studying this to understand whether hybridization changes their personality. So one of the standard measures of personality that people use is exploratory behavior. So with great tits in Europe, they do this by putting them in a aviary. They go out to Whiteham Woods and they grab a great tit and they bring them into the lab and they put them in this box, this room, and they fly around in the room and they measure that. So we, rather than bringing the birds to the room, we took the room to the birds. This is our little box on a trailer, and there's a bird. Oh, the video's working. Yay. So we're measuring how many times does he change perch inside the box. So we catch him in a mist net, put the cage on the side of the trailer, turn on the lights, let the bird go. It flies around in the box, turn the lights off, take the cage off, and he flies out. So it's a short-term, portable method for measuring exploratory behavior. And the pattern we get is, the graphic went away, but the blue is black-capped chickadee, and the red and other color are the Carolinas and the hybrids. So there's a measurable difference in the exploratory behavior of the hybrids and the Carolinas versus the black-caps. This is a kind of a messy plot, but the statistics support the idea that they are distinguishable. So we think that that is a reflection of a genetic influence on exploratory behavior, where the parent species differ in their degree of exploratory behavior. Black-capped chickadees are very confiding and mellow. I've had cases where I was putting a net up in front of a black-capped chickadee nest and had the bird stand on a perch a foot or two away from my face, waiting for me to get the net up so he could fly in the net. Meanwhile, if I did that with a Carolina chickadee, he would be 20 meters away screaming at me from a treetop. They are very different in their personalities, the two parent species. Well, that we've been able to show that the hybrids are kind of intermediate in their personalities. So we did this with exploratory behavior, and then we did it for boldness, which is another standard measure. So we did it for nest defense. So see if this video works. It's kind of fun when it does. I think it's working. So, oh, come on, keep going. There we go. So this is a coyote decoy that for some reason looks like a downy woodpecker, and the noise is a downy woodpecker. So that's actually a threat to a chickadee, because downy woodpeckers will take over their nest cavity. They can't actually get into the plastic ones, but the chickadees don't know that. So we're using that as a standardized stimulus to then measure. You saw the bird swooping down there. So how quickly do they respond? How aggressively do they respond? How persistent are they? To just quantify their boldness in defending their nest. And again, the black-capped chickadees are really bold, and the Carolinas generally are not. So we've got, and the hybrids are in between. So these are, and we actually, for this one, we actually investigated a single gene that's different between the two species. It's called DRD4. It's a dopamine receptor. So you can get into the weeds about the actual underlying mechanism that's causing one species to behave differently than the other species. We haven't done that much because that's a whole nother field of biology, but we've dabbled. It's kind of fun to play around with that kind of thing. I think I only have one or two more slides if this will proceed. Yeah. So I wanted to talk very briefly about the irruption birds and about winter behavior, because a lot of what I've emphasized so far is things that we do in the breeding season. In the non-breeding season, we focus on flock behavior, So this is a video. Is that going to run? Maybe not. This is one of our feeders with the antenna. So inside the lock and lock, the plastic box, is the circuit board that controls the antenna. When the bird lands on the antenna to come get seeds, the circuit board reads their tag. And what we get is a printout like this of the tag number, and then we look it up in our database and it tells us which bird was which. So if the video is running, you'd see different birds come in and the tag lights up as they land on the antenna. So what we do, so it's easy pass for birds, but it's in their leg band. What we can do with this is assume that if two birds come at the same time or close in time to the same bird feeder, they are social friends. They're associated with each other. If they never come to the same feeder at the same time and instead come 10 minutes apart and they avoid each other, then they're not associated. So we measure the birds that are together in a grouping as essentially becomes a flock. So here's a flock of each blob, each circle here is one bird, and the lines connect the birds in terms of how often they co-occur at a bird feeder. So these are chickadee flocks, and the red dots are the resident Carolina chickadees. So we can use this method to quantify the flocking behavior and the tendency for this flock to hang out at one bird feeder and this flock to hang out at a different bird feeder. And then we look at where are the eruption birds. And the short answer is the eruption birds are mixed in. So we found out early on that they actually stick around. They don't keep moving all through the winter. They come in and settle down and stay put for months. And nobody had that data point before. But we've had blackcaps stick around in the same place at Templin for almost two solid months. So while they're there, they're interacting with the resident birds. We had two extreme possibilities. One would be they'd be completely mixed in and just randomly mixed in with the Carolinas. The other possibility is they go their separate way. So there'd be a flock of blackcaps over here, and they wait for the Carolinas to get out of the way, and then they come into the feeder as a group. And the answer is sort of halfway in between. So they're mixed in mostly, although they too tend to be with each other, but they're not alone with other blackcaps. But they're definitely resident and hanging out, and then spring comes and they say bye and they take off and go back home, we think. We don't know where that is. If anyone ever sees one of our banded birds in Ontario, please let me know. Okay, the very last thing is our final master's project at Templin. Claire Jones thought that she wanted to use these smart feeders to study communication of the birds, because chickadee flocks are really important. The birds rely on getting along with their flock mates in the winter. So they're coordinating behavior with each other and cooperating with each other, even though they're also competing for food. But the cohesiveness of the flocks probably outweighs the competitiveness. Okay, so a smart feeder is a feeder that not only has the circuit board in it, the circuit board reads the tags of the birds landing on the antenna, but it also has a motor. So the circuit board can control when the feeder is open and when it's closed. And I didn't put it in here because we're only doing the work outside, away from Templin, but we're using this to study spatial memory in the hybrids up at Hawk Mountain, because we can program the smart feeder to only open for specific birds. So we assign, you go to feeder number 1, you go to feeder number 2, you go to feeder number 3, and we measure how long does it take them to figure out which feeder they have permission for. Okay, and the hybrids are really bad at it. And once they figure it out, they get stuck on it. So if you change it on them, they can't unlearn it. So that's a whole separate project. But what Claire did was use the smart feeders, not in conjunction with the bird's tag, but just the motor. So she's got, this is her scheme. The feeder is open in the morning, and then at 8 a.m., the door closes. And then at 10 a.m., it opens up again and stays open the rest of the day. Meanwhile, a second feeder nearby stayed open, and then it closed in the afternoon. So the birds don't know which feeder is going to be open when. And what she's measuring with automated recording units is the calls that they give just before the feeder closes, as it closes, and then as it reopens, and trying to measure, are they signaling to their buddies, hey, I just came to this feeder and there's no food, let's get out of here, so that they can all leave as a group. Or I just came to this feeder, hey guys, this feeder was closed a minute ago, now it's open, come here. This is where you want to be. So she's getting significant differences in the call composition in the open phase versus the closed phase. We don't know what it means, but it's consistent with the idea that the information that they're conveying in their chickadee calls is different depending on whether the feeder is open or closed. There aren't really any studies that have done it quite this way before, so this is pretty fun. This is the same chickadee call, but what people pointed out quite a long time ago about the chickadee call is that it's really special because the notes go A, B, C, D, 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 but they can vary how many A's, how many B's, how many C's, how many D's. They can leave one out. They can go A, C, D. They can go A, D. They can go straight to D. They never reverse the order, but they can change the number of notes in the sequence. So it has syntax. And so it's almost like a language. So there's a lot of information content. And what we're trying to do in this study is to quantify the information content. Are they conveying more information content when they find the feeder closed or when they find the feeder open? You know, is it more important to tell your friends, let's get out of here? Or is it more important to tell your friends, come here and join me? We don't know yet. So the jury's out on that, but that'll be our final project. I'm going to skip this. This was about just how people can help us out. And it's basically check our snags and see if there's any nests in them. This is my parting shot of a fledgling and his first flight. So this is me leaving my lab and going off to retirement. Because I am hoping to move on to do new things and not chase chickadees anymore. And with that, I'll thank you all, and thanks to French and Pickering Creeks for allowing us to continue to work at Templin. It's been a really good site over the years. Thank you for your attention. Sorry to run long, but
Questions?
Sure.
On your good nature for a few of those. We'll give you a chance to catch your breath and wet your whistle a little bit. I'll just a few quick things before we go into the Q&A. Just if you pass some signs, you probably saw we have our Iron Tour coming up the second Sunday in June. So now's a great time to register. It's still just $75 to register. So we encourage you to do that. Just go to our website. You can drive around and see all this great habitat. We have signs along the routes that show where it's been preserved, thanks to our easements and our awesome landowners who care about conservation. So providing lots of places for those birds to go in the future. And if you want to know about, if you're not already on our email list, there's a sheet there you can sign up to get notices about more programs in the future. So I'll turn it back over to you. Just give you a chance to catch your breath. Yep.
Yeah. During eruption, we've seen black cats in our yard. What we've seen is them like facing off in Carolina and back.
Yep.
Black cat, not very far away. I mean, within.
Yep.
Needs to go back. It's like a more aggressive version of the way chicks will say even be.
Yep.
It's more like get out of here, it's my territory.
Yep.
Is that a thing
Yep.
to observe? Is that what I think it is?
So your question is painful to me because that's one of the most basic parts of the whole hybridization story that I've wanted to know the answer to for 25 years and I still don't. And that is who's dominant, because what you're seeing is dominant subordinate behavior. There might be other things going on as well, but they do the dominance and subordinate things a lot at a feeder. And there are postures and displays that the dominant bird will do that signal that he's dominant. They generally puff up, and the subordinate birds will slick down and flinch. There was one study done in Ohio back in the early 2000s in captivity that found that if you put them together in a cage in an aviary, the Carolina males are dominant over the black-cap males. We've been trying to measure the same thing in the field, and it's been a really hard thing to measure. because it's very hard to get the Carolina chickadees to stick around at the feeder and interact with each other in close quarters, because you have to identify both birds. You know, you have to read all the color bands on both birds. We're hoping actually to go through the electronic data, the data stream, and look at the timing of when did the bird land on the feeder and did another bird immediately land on it right after them, to see if that's a displacement. But the challenge, and another study in black-capped chickadees showed that you could do that. The problem is you have to have video to corroborate it. So you have to have enough video to show that, yes, this is the right interpretation of that temporal pattern, but we're working on it. That's my other last master's student. I hope. Jim.
Yeah, who's gonna take over the nest after you retire?
Good question.
Seriously looking for.
Yeah, they're still out there. So I went around and checked about a third of them this spring, just out of curiosity, because one of the things I was interested in was to know how readily these Carolina chickadees would reuse the same snag, even though there was a nest in it last year. And the answer was they were really quick to do so. So that's good, because it means that they're making use of them, even if no one went around and clean, because we usually generally clean them out every winter. So it's not essential that you do that. Now, if there's a house wren nest in there from last year, they won't reuse that, because the house wrens just clutter it up with twigs and it's kind of off limits. So you do need to get the house wren nests out of there. But the older chickadee nest platform, they'll just put a new one on top of it.
The stag will stay there.
Until somebody from French and Pickering Creeks tells us to take them away, and they'll have to contact me up in Maine or wherever, tell me to come back and get them. Yeah.
Mother bird.
Well, I've been trying to, so Villanova hired a replacement for me, a woman who's going to work on marine fish. So she's not going to be checking chickadee nests. And I've been in touch with people like Dan Ardia at Franklin and Marshall to see if he would be interested. So I've been making other people at institutions in the area know that the system's available if they want to try to study it. But so far there's no takers. It wouldn't be the end of the world if we go for a few years without any data, especially for Great Marsh, where it's Carolina chickadees. The continuity is not essential. It'd be nice, but it's not essential, because someone else could pick it up again. And the most difficult part is the banding, because you have to have someone with the appropriate permits, and it's time consuming. So that there's a trick to that, but just checking the nests, anybody can do that, except if they're short. I've learned that the average height of the snags that I put out at the beginning of the study is not the average height of a typical student. So I got a lot of grief over the years about that. Take the top off. That's yeah.
I've been watching birds of different species at nests for a long time in a yard, and where you would expect one bird to always be dominant over another bird and chasing it away from the feeders. Doesn't seem to be the case, and so I'm wondering if there's another component in there, like how hungry am I?
Yeah.
How long have I been at the
Yep.
feeder, and I don't need to have this grab this apple.
Yeah. Yeah, that's definitely part of that. Motivation matters. So the bird state is a factor. And it gets complicated. So like at house finches, you know, everyone's got house finches. They're bird feeders. Who's dominant? The males actually let the brown birds have access to the feeder. They probably could be dominant if they chose to be, but they just opt not to. So they don't, the brown birds are more likely to go straight to the feeder and sort of push the red birds aside. Is it because they're more dominant, or because the red birds let them get away with it? Yeah, you know, so that's just one example of that's not exactly what you'd expect. Chickadees, we're pretty sure, black-capped chickadees are easier to study. You can go out in the winter in Ontario and in a morning characterize the dominance hierarchy in a flock of black-capped chickadees in one morning. That would take me a month to do it for Carolina chickadees, just because getting good observations on identifiable birds is so much harder. But if you put a feeding platform out in a place with black-capped chickadees, you'll get 3 or 4 of them on the feeding platform simultaneously. So you say, okay, he's dominant over him, he's dominant over him. Oh, here comes a 4th one, he's in the middle. Boom, 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 boom. It's easy. But these guys aren't easy. So it's just a fundamental difference in their tolerance of proximity of other individuals. Sometimes you get 2 Carolina chickadees coming to a feeder simultaneously, and you can see that one's dominant over the other, but you rarely get more than 2. And usually it's just 1, which slows everything down in terms of data collection. It's a challenge. Oh, by the way, if you see on the internet a picture of somebody with seeds in their hand, holding them out and having a chickadee land on it, it's a black-cap. I've actually asked the question on Twitter and Facebook, has anybody got a picture of a Carolina chickadee doing that? I've been asking that question for 10 years. Nobody. You can't do it. They won't come to your hand. So there's a challenge. I've tried. Yeah.
Yeah, first, I just want to say thank you for a lifelong career of amazing research. This
Thanks.
longevity study has fascinated me since I've been a birder. And your name pops into my mind all the time, just when I see.
Thank you. Appreciate it.
I logged a ton of questions. I'm going to reduce it down to 2 very simple ones.
Okay.
And the other ones I'll just speculate. So, but just with the eruptive black-capped chickadees, you had mentioned this mystery of where they go.
Yeah.
Have you guys pulled some feathers for hybridized stuff out?
We did.
Yeah.
Yeah, we didn't. It was an undergraduate project. Tried that.
Yeah.
I didn't want to get into it as a distraction.
Sure.
Her data weren't convincing to
Okay.
tell us very much.
Okay.
So there is a way to analyze the content of the feathers for what are called stable isotopes. And there's a signal where it varies. Things like hydrogen vary in the chemical composition. If you go really far north, you'll get a different signal than if you're in New York as opposed to Carolina.
Yeah.
So we tried.
Cool.
So it could be done, but
Yeah.
we just haven't done it enough to get a good
Yeah,
answer.
Sure. And then just a follow-up, I know these birds are resident, they tend to stick close to each other. Happen to be doing a bird survey in an area that's pretty close to here. So, just yesterday I was thinking of you because before this guy mentioned that you were speaking tonight, we were watching the end of the bird survey. We found ourselves staring at Carolina chickadees, just both parents running through a cavity.
Mhm.
Hilariously described exactly, it's this dead and dying,
Yeah.
this big,
Yeah.
Should I encounter a bird with color bands, should I take the time to document that? And if so,
Yeah.
who should I share it with? I mean,
That'd be great.
congratulations, you deserve to retire. Other than a brief-
Well, I would. So if you found a bird with a full set of color bands and you
Yeah.
could read all the colors, at Templin, I would be able to figure out exactly who it was because we don't repeat colors there. So each color band combination is unique.
Miles away, right? Or-
Pardon?
I'm doing a bird survey at Bentley Preserve, which is
Yeah, I don't
miles
know.
away.
That,
Do you think there'd ever?
that would be
a chance to
interesting.
check that?
So that's pushing the limits of, so the dispersal is very hard to study.
Right.
Because the average dispersal might only be half a mile, but it's the really important dispersers are the ones that go 5 miles, and that might be 1% of the
Right.
dispersers. But
Yeah,
if
I
they're
know-
successful,
probability is
yeah.
extremely low,
Yeah.
but,
we've had at Nolde where there's a lot more residents around the park. We've had people detect banded chickadees half a mile away. But most of our chickadees that we've banded at Nolde have dispersed within the park. So the average dispersal distance for a male is about 250 meters
Oh my goodness.
from their place of birth to where they breed.
That's-
In general, in birds, males stay home and females leave. So the average distance for the females is about 700 or 800 meters, which means we're losing some of the females because if they go sideways, they're out of the park. But still, it's not very far. But as I said, it's the tail of the distribution, the really long dispersers, which could be driving the movement of the hybrid zone and things like that.
Thank you.
Yeah. We had 1 or 2 reports of color banded birds in the Hawk Mountain area that were at least 2 miles away. Yeah. So it happens occasionally. Yeah. Jim, do you have another question?
Yeah, right.
And I want to make sure I have this right, that the Carolina chickadee is the less aggressive and less bold perhaps, or let's say aggressive. And the black cap is the one that had a lot of activity.
Right.
And
And yet the Carolinas seem to be the ones that are dominant over the black caps. So it just means that these measures of behavior don't necessarily translate. If you put a bird in a box and measure how it moves around, it's not necessarily translating into something that's meaningful in their normal affairs. Or maybe,
Bold and not be a jerk.
yeah, right, yeah. But the black caps are definitely more confiding than the Carolinas. But does that mean that they're easy to push around? I don't know. The Carolinas are just kind of jerks. The Carolina males are quite aggressive and noisy and, you know, complain a lot. Oh, I did want to say one more thing about the eruption birds that a lot of people find interesting about when do they erupt? Because they don't come every year. They come about every second or third year. And the story that seems to hold up is you get an eruption of seed-eating birds, not just chickadees, but also siskins and other northern seed-eating birds, when two things happen. They have to have a very productive spring breeding season, so a lot of young birds got born, and the subsequent summer and fall has a crash in the northern seed crops. So you got too many young birds relative to the amount of food. The young birds are the ones that come down. So when we can age the chickadees by the shape of the tail feathers, and the eruption birds are 90% of the time they're first year birds. So it's young birds that left home, wherever that was, haven't figured out whether they're speaking French or English. And they come down here, and as I said, they hang around for a couple months and then they leave. And whether they can track their way back to where they came from, we have no idea. The technology for tracking long distance movements is getting close to the point where you could put a transponder on a chickadee. They're doing it for some of the larger warblers now. So that would be really interesting to see. Where are they coming from, and do they go back to the same place, or do they just go north? Yeah.
So, have you used that motor?
No, I haven't.
For that?
No. And they've done it for, you know, swallow-sized birds, which are about 2 or 3 times as big. Chickadees are only 9 grams, so it's got to be.
Eastern chickadees, so we did a high eastern.
Kiwi, Towhee,
But they're bigger.
yeah. They're probably what, 30 grams, something like that. So about 3 times bigger. Yeah. But it's getting down there. I mean, people have done it for some of the larger warblers, like, yeah. Yeah, it could be done. We're getting there. I want the pit tags to get even smaller, because the ones that we used, they went from 12 millimeters long down to 8, but that's still too heavy. So we have some problems with the bands on the legs. You can put them under the skin, but then you have to do a surgery. So I've avoided that. But if they could get the pit tag down to like 4 millimeters, so it was only a little bit bigger than a normal color band, that would be nice. Yeah, that's been one of the limitations in that study, is that it's tricky to get it just right on the birds. If anyone's interested in some wonderful chickadee biology, you should look into the work that Vladimir Pravasudov and his group are doing at, he's on the list here, at Nevada Reno. They actually work in California on mountain chickadees. They have a high elevation population and a low elevation population. And the high elevation, we're talking really high elevation. They had snow last week, and they had to dig down through the snow to get to the nest boxes with the chickadees in it. So in that high elevation situation, the birds are very, very reliant on taking a seed from a feeder and hiding it somewhere and knowing where they put it. Whereas at low elevation, it's not as important. So they've actually measured a difference in their cognitive abilities depending on whether they're from the high elevation population or the low elevation population. It's just elegant work. It's work that 20 years from now is going to be described as one of the pioneer perfect studies. I'm blown away about. And we've tried to emulate some of their methods, as I mentioned at Hawk Mountain, doing the cognitive biology, and the squirrels said no. We'll shoot everything up. So, but it's fantastic stuff. So they publish about a paper every week. I'm very jealous, but very respectful too. Thank you.